Welcome to this explainer on genetic variations and vitamin A metabolism. Today, we're looking at how our DNA dictates the way our bodies process vital nutrients. Vitamin A acts as a crucial hormone like substance required for reproduction, embryonic development, cell differentiation, and immunity. We're going to see how minute genetic typos alter these biological processes, leading to severe health conditions. Biology is far from a one size fits all equation. Our agenda covers 5 key areas. 1. The carrot challenge. 2. The body smart switch. 3. The rechargeable eye battery. 4. When the blueprint fails. 5. The future of food. We're going to follow the sequential biological flow of nutrients from initial consumption in the gut through the bloodstream directly to the final destination in the eye. Section 1. The carrot challenge. Individual variation. Picture 2 people eating an identical amount of carrots. Do they absorb equal amounts of nutrition? Science shows the answer is absolutely no. Notice the difference between high converters and low converters. High converters easily process the beta-carotene found in carrots into usable vitamin A. Low converters struggle with this identical task. Their genetic variances limit absorption. The biological reason for this difference comes down to one specific gene in our DNA. Repeated genome-wide association studies confirm genetic polymorphisms alter blood and tissue levels of these nutrients. Some individuals possess an efficient genetic code, while others carry genetic typos. This brings us to the BCO1 gene. Think of BCO1 kind of like a pair of molecular scissors. The BCO1 enzyme symmetrically cuts a large provitamin A molecule, like beta-carotene, directly in half. Structurally, the enzyme resembles a 7-bladed propeller covered by a half dome, holding a central iron atom. This precise structural type produces retinaldehyde, the active form of the vitamin. If someone has a variation in the BCO1 gene, their molecular scissors are dull. They fail to cut the molecules efficiently. This failure leads to poor absorption, low vitamin status, and clinical conditions like hypovitaminosis A. Section 2. The body smart switch. Preventing toxicity. The human body must avoid accumulating toxic levels of dietary lipids. This necessitates the ISX smart switch. First, the body detects sufficient vitamin levels inside the system. Next, a homeodomain transcription factor named ISX activates. ISX actively suppresses the genes responsible for absorption, specifically SCRB1 and BCO1 inside the enterocytes of the gut. As a result, the gut actively stops absorbing beta-carotene. This negative feedback loop perfectly prevents toxic attenuation. It acts as a flawless biological safety switch to manage fluctuating dietary supplies. Section 3. The rechargeable eye battery. How we see. Notice the continuous molecular loop of the visual cycle. When light hits the eye, it bleaches a chemical called 11-cis-retinol bound to opsin proteins, initiating phototransduction. After this bleaching event, the eye must recharge the chemical to sustain vision. Specific enzymes process the bleached molecules and loop them back into their active state. Without this continuous cycle, human vision ceases to function. Picture this as a rechargeable battery with a precise timeline. In milliseconds, light bleaches the compound from 11-cis-retinol to all trans-retinol. After bright light exposure, this photoproduct exists in millimolar concentrations. Over the next few seconds, RDH enzymes rapidly clear away these resultant toxic photoproducts. If uncleared, they form dangerous side products. Within minutes, the RPE65 enzyme takes the cleared compounds and recharges them back into 11-cis-retinol. Without this continuous recharging phase, vision fails rapidly. Section 4. When the blueprint fails. Clinical impact. Specific clinical impacts arise from different mutations. Mutations in the STRA6 gene cause Mackey-Wood syndrome, resulting in severe birth defects like heart anomalies. Variations in RPE65 or LRAT lead to Leber congenital amaurosis, a form of childhood blindness caused by a failure to recharge the eye. Finally, BCO1 variations limit vitamin production, causing night blindness. Every single row represents a different genetic typo resulting in a severe health consequence, highlighting the absolute need for internal biological maintenance. Let us look closer at the STRA6 protein acting as a cellular doorway. STRA6 facilitates the bidirectional flux of nutrients across the cell membrane, allowing the vitamin to enter from the bloodstream. If this doorway is broken, the cells starve. The results are severe birth defects, including pulmonary dysplasia, cardiac defects, or infants born completely missing their eyes. A single broken doorway yields devastating developmental failures. When the eye fails to recharge its visual chromophore due to a mutation in the RPE65 gene, the cells experience continuous activation by an unliganded opsin. This error directly causes the slow, progressive death of the rod cells in the eye. Research estimates mutations in the RPE65 gene account for up to 16% of Leber congenital amaurosis cases. It perfectly illustrates the fragile nature of cellular health and the absolute necessity of a functioning molecular battery. Section 5. The future of food. Beyond one size fits all. If our DNA dictates nutrient absorption rates, is a single universal dietary recommendation logical? Given the genetic variations we examined in enzymes like BCO1, blanket dietary advice ignores fundamental human biology. Individuals process identical foods with vastly different physiological outcomes. The evidence points to one logical conclusion: personalized nutrition is essential. High converters and low converters process food differently. We must account for genetic typos in enzymes like BCO1 to ensure proper health outcomes. Are you eating a diet designed for your unique genetic blueprint? Or are you hoping a universal diet meets your specific biological needs?